All right. So our first talk today, Jing Huang is a PhD student at Mississippi State University's Department of Wildlife, Fisheries, and Aquaculture and the Thad Cochran National Warmwater Aquaculture Center. Under the guidance of Dr. Fernando Yamamoto, Jing's research primarily focuses on enhancing catfish nutrition. Specifically, his focus lies in identifying optimal probiotics for catfish to improve health and growth. So go ahead and take it away.
Okay, and thank you for your introduce. Good afternoon, I'm Jing, the PhD student of Mississippi State University. Today, I'm going to talk about some of my research I have done so far of my project, which has been looking for powerful probiotics for the hybrid catfish. First, I would like to briefly introduce the background of my study. As you may know, catfish is a primary cultural species in the U.S. aquaculture industry. Mississippi, Alabama, Arkansas, and Texas are the top 4 states in catfish productions, which contribute 97% of the total sales in the U.S. However, numerous catfish pathogens are shown in the pond, causing a serious impact on the industry and economic loss. Here are some of the cases in a bacterial infection on catfish. Coronella disease, NAS, and ESC are 3 of the major diseases caused by bacteria. The corresponding pathogens are shown in the right here. To solve the problems, antibiotics are generally applied as antimicrobial agent for disease treatment in aquatic animals. The following table here lists out the current FDA-approved antibiotic and antimicrobial agent for aquaculture production in the US. And antibiotics are powerful and effective in most of the bacterial infection cases. However, the use of antibiotics not only increase the production cost, but also do the selection work on the local microbial and pathogen. The misuse and overuse of the antibiotic leading the drug resistance develop and transmit within the bacterium populations. So here comes the questions. Is there any alternative way to lower down the use of antibiotic in aquaculture? So the answer is yes. The following picture here shows the alternative strategy for reducing the use of antibiotic. There are 4 popular ways in the pictures. The selection of the cultured fish, developing vaccine, antimicrobial, or providing functional feed with immune stimulate, probiotics, or any related supplement. Probiotics are groups of live microbiome living in the digesting systems or the outer surface of the organism, with a positive effect to the host. The functions of the probiotics has been studied in multiple aquatic animals and applied into the aquaculture industry as well. Although different microbiome may invoke various effects, probiotics are generally have the functions on improving digestion, absorptions, antimicrobial, and immune stimulation abilities in the aquatic animals. So for the goal of this study, we have 2 objectives in these studies. In objective 1, we are trying to isolate potential probiotics from a hybrid catfish and evaluate by proteolytic and lipolytic activities and biosafety assessment. We will further investigate the probiotic traits of the isolate candidate in the objective 2. So in the objective 1, we start from the investigation of the microbial community in the local cultural hybrid catfish. It's common to find the fish have the size disparity problems in the same cultural environment. And these phenomena are generally regarded as the individual difference in the past until the effect of microbiota to their host is studied and discovered. Based on the idea, we hypothesize the intestine microbiome might be one of the key factors affecting their natural growth performance. The fish from three surrounding ponds are categorized into overperforming and underperforming groups based on their average body weight and the length of the populations. To collecting the digesta and analyzes the intestine microbial compositions, fish cultured in the earth pond are fed with commercial diet, and 20 hours after feeding, we harvest the fish and collected their digesta samples for DNA extraction. Extract DNA from the digesta were subject to Illumina mycexistence and target on 16S ribosomal RNA V4 region. The sample were sequenced by University of Minnesota Genomic Centers. Afterward, the sequence data were analyzed on CLC Genomic Working Bench following the instruction from the developer. The reads, which passed the quality control, were assembled and clustered into outs and further identified by using a bus with NCBI database. The digesta of hybrid catfish were collected for microbiota analysis to verify our previous hypothesis. So first, we analyzed the sample microbial abundance by alpha diversity. From the figure 1A here shows there's no significant was found in the alpha diversity, which means no significant abundance difference in the sample. Next, the diversity between the populations was checked by the beta diversity analysis. Something interesting here we found from the beta diversity analysis, a part of outperforming samples are separated out from the underperforming fish. The significant difference between the groups were found from the post-test, which indicates some overperforming fish are having different microbiota comparing to the underperforming fish. So from here, another question comes out: do we hit the target? Is the microbial composition affecting the growth performance of the fish? So we keep on analysis and look into the relative microbial abundance. And we notice there are three genus shows a trend on change between the overperforming and underperforming fish, which give us a hint. Those bacteria might related to the growth performance of the hybrid catfish. As the figure 2 here shows, three genus of bacteria have a change in abundance. Overperforming fish has less cetobacterium but higher in Letococcus and Turicibacter. According to the results, we target on Letococcus for the following selection. So based on the microbiota analysis result, we start the isolation and focus on extracellular enzyme secretion and antimicrobial ability as a primary selection standard. The selection of bacterium candidates will follow this order. Unqualified isolate will be removed. The biosafety assessment were applied to ensure the safety of the probiotic candidate to the host. We will screen the isolate and do more investigations on the biochemical characteristics of the isolate to have extra information for the future applications. So in these studies, in total, 42 bacteria were isolated from the digesta of the overperforming fish. To isolate the bacterium as hybrid catfish probiotic, we keep some digesta samples from the microbiota analysis and trying to isolate the target bacteria from the overperforming fish. First, we apply 3 mL of the saline buffer (PBS) to the preserved digesta samples, and both tests take out 1 mL of the supernatant. After that, a tenfold serial dilution was conducted before inoculate the suspension on Lactobacillus culture medium (NRS) and the NRS modified, as well as non-selective isolation medium (TSA) will be used in this study as well. To mimic the environment in the fish intestine, we incubate the sample in 27 degrees Celsius overnight. As the picture here shows, the bacterium colonies were demonstrated on the culture plate with different morphology. All the interest colonies were picked up and subject to the purifications. The isolate were preserved in -80 with 20% glycerol supply. To testing the extracellular enzyme secretion ability of the isolate, the purified isolates were inoculated into corresponding broths and incubated for 24 hours. We centrifuge the bacterium suspension and apply the supernatant into a pre-drilled 8 mm well. Incubate overnight and let the testing supernatant diffusions on the agarose. The whole alone were measured to identify the digestive related enzyme secretion ability of the isolate. So as a result here, in proteolytics and lipolytic ability test, the testing is modified from the plate diffusion method, which we mentioned in the previous slide. This method gives an advantage on rapid screening of all the isolates. Testing plates were enriched by adding extra 1% of the semi-milk for proteolytic and 1% tribulin for lipolytic ability test. Within all the isolates, there are 20 isolates demonstrate a better extracellular enzyme secretion abilities, shows in the figure 3 here, reveal a higher possibility on improving digestibility after we subject them into the host intestine. In the antimicrobial ability test, 3 catfish pathogens causing the NAS and ESC were applied in this test. Same as enzyme secretion ability test, the pathogen bacterium were pre-separated onto the BHI medium before inoculate the bacterium suspensions in the pre-drill hole. The process was shown in the picture here. So after incubate for 24 hours, 20 isolates demonstrate antimicrobial ability on 3 catfish pathogens. As another important selection standard, the growth of the testing pathogen was inhibited by the applied bacterium, give the advantage on prevent the infection or against those pathogens to the catfish. Biosafety assessment is another important step before we introduce the probiotics into the cultural species. In this study, 8 common use antibiotics were applied in this test. All the isolate has no hemolytic abilities and suspect to at least 1 antibiotic. To well understanding the metabolic profile of the isolate, the biochemical characteristic API-20 was investigated. All the isolate past the selection standard are identified as gram-positive bacterium with no significant catalyst abilities demonstrated from the results. All the isolates were subject to 16S ribosomal RNA sequencing. If the results match to any previous report pathogens, the isolate will be excluded from the candidate. So after comprehensive considerations, NA5 was standing out from all the candidates and identified a Staphylococcus species from the 16S ribosomal RNA sequencing. In the objective 2, we focus on the best isolate NA5 and analyze their coding genes by using whole genome sequencing. The following are the resources we use for analysis and the related results. So in silico analysis, given the outline of our interest bacterium, the isolate candidate NA5 was identified as Staphylococcus lattice with high similarities. After that, the possibility to be human pathogen was assessed by searching the variance genes in the NA5. We received the negative feedback from the match, confirming NA5 is not a pathogen to a human and mammals. Among the input genome, 28% of the coding region were matched to an NCBI library and subject to 28 universal functions. The functions with over 50 features were displayed in here, demonstrate the relative function pathway of the NA5 from the coding genes, give the outline of our predict functions. To further understanding the functions of NA5, probiotic trait was investigated in this study. We categorized the identified coding genes into several features, which consider benefit to the host. Predict functions of the NA5 are including amino acid metabolism, lactic acid productions, intestine adaptations, digestive related enzyme secretion abilities, and vitamin B biosynthesis. Notably, multiple vitamins, including vitamin B1, 2, 6, biotin, and 9, will be able to produce by the NA5, providing the extra nutrients for the cultural species. Moreover, from the intestine adaptations, temperature, acid, biosalt, and osmotic oxidative stress tolerance abilities was found, which indicate NA5 has a robust survival abilities in the intestine of the aquatic animals. In the bacteriocin productions, 3 bacteriocin productions was predicted from the whole genome of the NA5. The discovery of leucocosin A producing in NA5 giving the advantage on maintain the purify of NA5 after we trying to deliver it into the host. So the final is the conclusions. In this study, the intestine microbial communities in the hybrid catfish was investigated. Leucococcus species has a higher relative abundance in the overperforming fish. A potential probiotics was isolated with proteolytics and lipolytic abilities, benefit to the digestion of the host. In vitro antimicrobial tests prove the possibilities of NA5 against the pathogen of NAS and ESC disease. The isolate candidate was considering have lower risk and not a pathogen. From the whole genome sequencing, NA5 was identified as Leucococcus lattice. Several traits were further identified, giving an advantage to the host by providing extra digestion enzyme, amino acid, vitamin B supplementations. The secretion of lactic acid and bacteriocin might help the host prevent the embedding of pathogens, but the extra test is required for verify this hypothesis. Here's some of our future direction for these studies. Here, first, we will identify the best medium to increase the survival rate of NA5 after the lymphodylation preserve. And the probiotic dry powder will be mixed with the commercial diet for feeding trial. We will also want to know which lab stage of the hybrid catfish may have the best effect after receive this probiotic. If we have a positive feedback from the feeding trial, a large-scale testing and standard operating produce SOP will be conducted. We hope this study can give the U.S. catfish culture industry a new but reliable options on functional feed. This work is funded by an AFS-FHS grant. I would like to thank all the help from my advisor, Dr. Frendo, and co-advisor, Dr. Jordan, and all the suggestions from Dr. Kathleen, Dr. Matt, Dr. Allen, and Dr. Weiss, and also my lab mate, Penny, Grant, Sandro, Bia, Thiago, and Marcus on their supporting and helping during my studies. Finally, I would like to thank you for all your listening and participate. Thank you.
Well, I have a question for starters while we're waiting to see if anybody else has one. What do you see as one of the biggest impediments or problems taking a probiotic like this and moving it to a more large scale commercial use? Like, what do you see as some of the kind of roadblocks that, if your work shows that this is promising, getting it out to farmers and producers, what are some potential roadblocks that you might run into with that?
Okay, thanks for your questions. Well, for these questions, I think it's a little bit tricky. As some of people may know, probiotics is always working very well, or most of them is working very well in the laboratory conditions. I mean, in the recircular systems or in the aquarium tanks. But the problem is, if we're trying to apply the probiotic diet into the, as the picture here shows, the earth pond or in the large scales, and I think they will have a lot of factor will jumping into here and maybe affecting to a result. So I think the most challenging things for up to the large scales will be a way of feeding control the aquaculture management, and also to increase the survival rate of the probiotics when we mixing a feed and apply to the pond.
Thank you. Does anybody else